Hello, good evening, everyone. Good evening, very distinguished colleagues, and welcome to the special edition of the Clinical Pharmacists Association of Nigeria weekly clinical meeting. My name is Dr. Onyime Shet, and I am the moderator of today's presentation. So before I begin to recognize VIPs, I want to respectfully invite Pharmacist Lawal Mohammed to lead us in a short prayer. Hello, sir. Hello, sir. Hello, sir. Okay. Hello, sir.
Hello.
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 Please,
I can hear.
Lawal, lead us in a short prayer before we start.
I can hear you.
Okay, let's pray. Oh God Almighty, we thank you for the gift of life. We thank you for the gift of strength. We thank you that we are able to gather here today. That will affect our practice positively. We therefore ask for your presence. Lead us this clinical section, so that at the end of the day, we give your name all the glory. In Almighty God's name.
Amen. Doctor, can you unmute yourself and continue, please? Only unmute yourself and continue. That's it. Kindly unmute yourself and continue, Dr. Izet.
Okay. Okay, thank you very much. I welcome you again to today's special edition of CPAN Weekly Clinical Meeting. Please permit me to recognize the presence of fellows of the Pharmaceutical Society of Nigeria, members of the National Executive Council of PSN, members of NEC of CPAN, fellows of our prestigious West African Postgraduate College of Pharmacists, I also welcome and recognize DEX officers and ESCO members of the various clinical pharmacy specialty groups and members of CPAN in Nigeria and in the diaspora. Ladies and gentlemen, you are all highly welcome to today's clinical presentation. The topic for today's presentation is basic test for medication safety and efficacy in clinical settings. And our presentation we have like 1 hour, 30 minutes for this presentation. We will have taken 45 minutes for the presentation itself, then about 30 minutes for questions or comments. Then the remaining 15 minutes will be closing formalities. Our presenter today is Dr. Sunday Olajide Awofisayo. Dr. Awofisayo is a molecular biopharmaceuticist and a clinical pharmacokineticist. He is a laboratory analyst and has been licensed to analyze food, water, drugs, cosmetics, medical consumables, and agro-allied products. Dr. Awofisayo is the head of the department of the Department of Clinical Pharmacy and Biopharmacy in the University of Uyo, Nigeria. He is the principal investigator and chief executive of Bioscientific Group. He is also a licensed public analyst of Nigeria. Dr. Awofisayo has over 20 years experience in training pharmacy students. Is involved in the training of undergraduates and postgraduate pharmacy students. Please, let's welcome to the stage Dr. Awofisayo to do justice to the topic. I am sure you will all learn a whole lot today. Thank you. So we welcome Dr. Awofisayo to come and present the basic test for medication safety and efficacy in clinical settings. Thank you, sir. Welcome, sir.
Good evening, distinguished practitioners. Ladies and gentlemen, all protocols duly observed. My name is Dr. Sunday Olajide Awofisayo. It's my pleasure and a big honor to be asked to give the talk for this evening. I believe by God's grace, the lecture will be quite stimulating. And of course, I expect a lot of questions, contributions at the end of the presentation. And so I don't want to waste our time. Let us go right into the presentation. Basic tests for medication safety and efficacy in clinical settings. This is our outline for this presentation. And I have a preamble for us this evening, for us to just look at before we go into the lecture proper. The first one says, achieving safe and therapeutic concentrations of drugs is a target in clinical settings. I make it a point of duty for my students from year 3 to note that when we take drugs, we actually want the drugs to get to a concentration in our system where it can work. It's either it gets there, it doesn't get there, or it gets beyond where it should be. When it gets beyond where it should be, we're going to see a whole lot of toxicities. When it doesn't get there, then we won't really get the result we're looking for. Okay? I'm sure we are quite conversant with the concentration time plots. Minimum effective concentration, minimum toxic concentration, and whether it is oral, intramuscular, and all of that, the way the graphs go. And all we want to do is to keep the drug at that therapeutic window. Fine. Now, the second bullet there says quality can only be built into therapeutics or clinical practice, but it can't be inspected into it. So before we start talking about minimum effective concentration and all of that, we need to be sure that we have quality drugs with us. And so if any other group of persons wants to be interested in quality drugs or the authenticity of drugs, it should be clinical pharmacists. Because if the drugs we have in our hands or that we are talking about are not what the label claims say they are, then we are just joking. And so at this current time, at this present dispensation, we have issues a lot with the quality or the quality of the drugs we have on our hands. And so the next bullet says foundations matter most. So we need to be sure that we get it right from the foundation. Quality drugs are basic. There's no compromise about it. They must be what we think they are, or we believe they should be. And if anyone in the healthcare team does not get this and does not give attention to what we're talking about, then we are going to have to forget about the entire stuff. So let me go to the introduction to basic tests. What are basic tests? These are simple identification quality tests that should be adopted foundationally on drugs, especially in all clinical settings. I say all clinical settings because I know and I'm aware, and in fact, most of my publications have actually pointed at the quality of drugs. And so basic tests are performed on both the active pharmaceutical ingredients, especially when we are in the industries to manufacture. And then outside the manufacturing industries, the drug products keep moving from one point to the other. And I think I need to make a submission this evening that the drug products for clinical use ought to at least this basic test for their identification, their authentication. This test may precede the request for thorough analytical test if we have a reason to think this might not be the drug we have, that is labeled. I want to also challenge us that if we have to go to different pharmacy outfits or even some hospitals and look at the drugs we have there that we intend to use for patient care, we won't be surprised to see quite a gamut of them that are not what they are labeled to contain. We're not talking about the quantity. We're actually even looking at whether that ingredient is there. It might not be there. It might be something else. It might just be ordinary blank. Sometimes we use blank to bring down blood pressures. We use blank to control blood sugars. And when we don't have the outcome we're looking for, we are either looking for some other reasons to explain why it is not like that. So basic tests for drug products are most suited for clinical pharmacists as a matter of safety and efficacy. Safety as in, if it is not what it is labeled, as in if there is another chemical entity packaged in the name of something else, we cannot exactly tell the safety implications of those stuff. And most of us in our practices, we give out these drugs believing that that is what it is. And it might not be. So for us to give meaning to all the pharmaceutical care interventions, right from the first to the last. We are saying this evening that the drug we have in our hands or that we are talking about have to be what they are labeled to be. So what basic tests are very simple. They require 1, 2, or 3 steps to actualize what we want to do. It's a very simple protocol. It's only we have not paid attention to them. And we don't even know at what point the attention should be emphasized. But I am saying this evening that even in community practice settings and in hospital practice settings, where we have most of our members, clinical pharmacists, we will need to start from the scratch and be sure that the molecules we have or the product, the product we have in our hands actually contain what the label claim says they contain. So basic tests don't have any high throughput. It can be that simple. And as simple as these steps are, if pharmacists don't do these tests to confirm that these drugs are what they should be, before we move to higher levels of our care, then who will do it? I want to say this evening that if we don't look in this direction, in the light of the noise all over the place, fake drug, this, that, and all of that, somebody else will start doing it soon or later. And we're talking about drugs here. That's our domain. That's our constituency. So I propose at this junction that a unit in every hospital, every community practice should, hold a basic test unit where we can, after collecting a consignment of paracetamol tablets from EMSO or from M&B or any company at that, we just randomly select 1 or 2 sachets and then remove 1 or 2 tablets, crush them, and then do a little confirmatory or identification test to be sure that this should or is paracetamol. If what we do is just to arrange them on our shelves and start dispensing them, I think we have omitted something very vital, something important. So I posed a question to the House this evening. 1, do we wait till some other scientists start doing things like this? And then we now start telling them, no, that is drug. We are the ones to do it. I don't think we should wait. Then are the other clinical team members looking up to pharmacists in this regard? I think so. Because if we have a team, a grand round of consultants, this, that, and then consultant pharmacists, this, that, and then we have drugs to talk about. When it comes to drugs, they want to look at us. Pharm or consultant, this drug, are we sure it is? Are we just going to nod our head? Yes, I think we got them from the right source. Or getting drugs from where we think is the right source might not be enough to say that the drugs are original. We live at a time that people can kill people because they want to extract some parts from their bodies. This same set of persons can do anything to drugs along the distribution chain. I'm aware that people do, even reps can stop at Onitsha or Aba and do their abracadabra on, because they just want to make it fast, rich, quick. And when these drugs come to us as the practitioners, we just go ahead and use them just the way they are. Then I think there's something that we need to do extra. So these tests are inexpensive. We need limited resources and reagents to do them. And the training we had while in school are just enough to help us achieve what we want to do. So no expensive instrumental setup is required. So we are not going to tell our employers, we need this mass spec, GC, NMR. We don't need those high-sounding equipment to do basic test, basic test. We don't even need big space or facilities to even do them. I have some questions here again. Won't it be a thing of pride if clinical pharmacists are known to usually validate and authenticate the drug products? If we give authorization to other healthcare workers that this drug is captopril, this drug is enalapril, these are angiotensin converting enzyme inhibitors. So since the doctor has requested that we use this in these patients, you're handing over to them. And then having used the drug at the right doses and all of that, we want to follow up on, and then want to see what the outcome is. But the drug we're putting in the hand of our co-healthcare team members, we are not even sure. So it can surprise us as well as how it's surprising them that the drug actually entered into our facility through this, and they want to start looking at who supplied it, who did this. Oh no, I think we need to pay attention to how to nip these fraudsters or these sharp practices in the board as part of our practice, because that's our tool. If we don't sharpen our tools, then it's not going to cut the tree. If we need to fall a tree and the knife is not sharp, we'll soon find out that we're wasting a lot of time. Okay, then I dream of a system where all health service providers, and indeed everyone, the patients, will be looking up to the pharmacist for the authenticity, for the validation of their drugs. Today, I make it bold to say that if the same person that supplied XYZ pharmacy goes to supply a patent medicine or any other person who is interested in drugs, and all we do is display them on our shelves, and we are not going to let the people know that we do some extra scientific checks to be sure that these drugs are what then I don't think we're doing anything too different. And so we need to start looking in this direction and bring out something that will give us recognition. So application of basic tests. Yes, in community setting, in hospital settings, I propose that there should be a unit in these two. Every clinic, every pharmacy outfit where a clinical pharmacist operates. Before he gives all the drug information and make all the pharmaceutical care interventions and all of that, let him include that this drug that we are talking about, first, are what they should be. We are not going to say we don't know that there are fake drugs all over the place. I won't be too unsure to say that there are more fake drugs than we have original ones. Because the distribution chain, the network is still very faulty. And we're not going to that area this evening. So we claim we are responsible for drug and drug-related issues. They let us take it from the authenticity, the genuineness of every drug, starting from paracetamol to metronidazole, flagyl, to every drug at that. I have some questions here again. Just for us to, a kind of illustration. Why do banks have to put UV lights on every currency that is coming in? Because they want to ensure that if Mr. X comes to withdraw 50,000 naira, all the notes that he goes back with are original notes. Somebody could have brought some counters to deposit, and the bank also just packs it, stamps it, and gives out. If we keep doing that, or collecting counterfeit notes from banks, then banks will cease to become banks. So, as clinical pharmacists, because drugs are what we are deliberating or talking or making all our attention on, they must be original drugs. So we're going to carry that one as a baggage for our travel. If we leave that one behind, someday, sometime, somebody might embarrass us. And they even believe that pharmacists are the ones that are making drugs. Yes, they know that drugs are our products. Then indeed, we want to look for those ones that are not and say, please, there's one batch of whatever that I've just tested, communicating with one another, giving signals. Why don't you try it and see? And before we know it, all those people are doing all these fake businesses. We know that it can't be business as usual. We move to the next slide. I have samples of Quartem here. Just a few days ago, somebody brought the fake one, and I had to see the original one. I was able to see the difference, really, even though I've not done any basic test, as it were. But the truth is, this person, the pharmacist from whose shop the fake Quartem was bought, also still has the original. Everything was just mixed up in that carton. We have original ones there, we have the fake ones, everything just mixed up. And all he's doing is just selling. Well, this person is not a clinical pharmacist. But I'm making a proposal this evening because clinical pharmacists are more serious about the outcome of drug therapies on patients. And I think we should pay attention to the genuineness of products. I make it bold to also say that even the fake ones look more sharp. They are sharper than the original ones. And not just by visual inspection. We will need to do one or two simple things, which I know as we continue to do it, it gives us some pride. It gives us some confidence. It gives us some boldness. I'm a pharmacist, I'm doing something. That's what I think. So I have two other products here on this slide here. The first time I was going to travel outside this country, or the first time I was going to enter into an airplane, my university was sponsoring me to Japan to make a presentation on basic test program that I developed for Viagra. The Pfizer product on our left here is Viagra, 100 milligram. That was 2006. As at that year, original Viagra sells for 6,800, 4 tablets. And there was preponderance of the fake ones all over the place, so for 2005. And so if you don't want to sell the fake one, still with Pfizer, carton, and all of that, then be prepared to just keep your original on your shelf. People are not buying. So the question here is, the price difference moves the fake one. I won't call it fake or have the authority to call it fake until I did the basic test. So I did a basic test. I bought the 2005 and the one of 6008 and did a simple test on them and found out that the outcome on the original one, the fake one didn't have it. What I didn't go ahead to do is to find out what exactly is in that fake one. Because if people keep buying it, could it be that it's also still giving them a result? Or they're still buying it because of that cheap price and they use it and it's not giving them a result, and they're thinking their condition is beyond what drug can handle. And of course, in Nigeria, where we have about 250 million persons, until the fake drugs are completely exhausted, I think the price differential will still keep the sales of that fake one going. And so I did a little test and put it on the internet. I was invited by Elsevier Tetrahedron, and I understand that's the highest body of organic chemists, to present what I have done in Kyoto, Japan. And so my vice chancellor was so excited. He gave me money, $150 every day to travel out for my feeding and everything. They paid the air travel up, down, and up. And it opened my eyes to quite a world of opportunities. And so what I did there, I've also published it. And the journal is on the reference section of this presentation. And so we will need to do tests like this to be able to take the wheat from the chaff so that we can have results. Otherwise, all of this kind of stuff will keep messing us up. So the basics of basic tests. Number one, the alarming rate of counterfeit and substandard medications across the globe. All this pharma fraud. And not even the big pharma, even everybody's interested in drugs now. And people want to just put blanks and label them and do all of that, okay? Then we have a lot of previously adopted faulty methods to checkmate this trend. We've used the computer code stuff, send a message to, and those things are just not working. And so what we need to do, stemming from the basic test, is to impregnate some strips like litmus paper that can make basic tests more simple, even for people out there to even try them out themselves. If a paper is impregnated with some chemicals, we only need to remove a tablet, sacrifice one tablet, crush it, add water or whatever to it, dip this strip we have supplied. So it will now become a problem for those who are going to fake the drug to also make their fake strips that will give the same result with the original. And people can now say kudos to pharmacists for coming up with, because like I know, everybody is already tired of this issue of fake drugs. Only God knows how many people have died from, or who should have not died if they had the right drugs in their hands. And so this is the whole importance of biopharmaceutics. I'm happy to be a biopharmaceutics, okay? And I know that study has to do a lot with the physicochemical properties of drugs. I make it compulsory for my students from year 3 to have on their fingertips some basic parameters or properties of drugs like metronidazole, flagyl. Every clinical pharmacist, as it were, should know that this drug is a basic drug. And so because of that basicity, or the pKa or pKb and all of that, it behaves in some particular way in our system. The absorption, the Ka absorption, the TF, the everything, all of those things we're talking in pharmacokinetics have to do with the physicochemical properties of drugs. And I want to think, if we are doing seminars, we should call it pharmaceutical care case studies, not just a case study. I'm familiar with some case studies around me, organized by pharmacists. They call it a case study. And the content of that case study is not highlighting physicochemical properties of drugs and the disposition of drugs. So that if a or a lab scientist or a nurse comes around, they hear something new. Most of them don't even know if NSAIDs are acidic drugs. And if an acidic drug is taken orally and it gets into the stomach has a pH, an acidic pH. And then we now talk about whether it is going to be ionized or unionized. And that affects the absorption and all of that. So we need to talk about that for every drug so that they can listen to us. If we keep talking to them at their own biomedical arena, they were not going to make a lot of impact. That's my belief. And so we need to take the battle or take our contribution to a level. Eagles, when they want to fight a snake, it picks the snake from the floor to the sky, where the snake is lost. Doesn't even know how to, until the snake dies, and then it comes back to come and eat it up. But if the eagle wants to stand on the floor and start fighting the snake and all of that, I don't think it will be an easy battle. So drugs, physicochemical properties of drugs and all of that is our domain. And this basic test is still hinging on the physicochemical properties of drugs. And we can use that to tell which is okay and which is not okay. And then some drug products come to us, we don't even know the excipients they have used. Because I also think the excipients that have been incorporated into that drug can also affect a lot of outcomes of the drug. And this, our colleagues in the healthcare team, they want to listen to us. We need to say something. So when we are saying something, let us talk to them from the selection of excipients and the release rate of the active ingredient and all of that. If we keep talking about pharmacology, that is still a domain. I'm not saying we're not going to talk about pharmacology. We're going to talk about pharmacology. We're going to start from the chemistry of the drug, the interaction, the stereoisomerism, all those things it will give us credibility and relevance. So all the inherent functional groups in different drugs, we need to visit them again. And because you can hear a pharmacist saying, I don't like chemistry. That is a core. And so if the excipients are unknown, we may have to extract the API from the drug product. And it's also a very simple protocol. For example, if I just remove one paracetamol tablet, crush it, dissolve it in acetone, filter through a filter paper, all the excipients will be on the filter paper. The acetone that filters through will carry the paracetamol molecule. I can allow the acetone to evaporate and scrape my paracetamol form, and then do 1 or 2 basic tests, whether I'm going to add sodium hydroxide, just 1 or 2 mil and all of that. And then I want to see whether all these test tube things, yeah, and this is our area that we think or we don't even pay attention to. And if we start doing that, we're going to save a lot of lives. And then the pattern of presentation of our observation of outcomes will actually change. And if these other persons in the healthcare team, we know that we do this for our drugs, then they're going to come asking, please let me find out whether this is okay, whether this is not okay, and all of that. Because as it is now, it seems that we need to work on how to make our recognition. And so my concept of clinical pharmacy, I just decided to use this definition so that we can juxtapose it with whatever form definition you have for clinical pharmacy. I'm saying it's a branch of pharmacy or pharmaceutical sciences that deals with the utilization of pharmacist knowledge, skill, judgment. And all this knowledge, skill, and judgment are an embodiment of all the different departments we have in pharmacy practice. Pharmchem, pharmacology, pharmaceutics. We need to bring everything together as a clinical pharmacist to be able to fly. If we are looking at clinical pharmacy on its own, isolated, no input from all these other hands, then we're not going to be able to make a lot of impact, as it were. So I'm believing that we need to, come from those fronts and present a formidable a position. Well, it might be that in America, basic test is not popular. Yeah, their situation there, their condition in America does not even warrant that. They have a very structured drug distribution network. So here that we don't have it, we can't just take what they are doing there, hook, line, and sinker. We will need to adopt or formulate and put our own structure in place alongside with what they are handing down to us so that we can have a better result for our own environment. So this is very situational. It's environmental. And so the definition I have there, I'm sure we can all see it on the slide. So we're going to be putting biomedical skills, judgments, knowledge, biopharmaceutical skills, knowledge, and other affiliated areas in place to present a curriculum or a structure or a modus operandi for clinical pharmacy practice. In the case of drug-related patient management, authentication of label claim should appear as first in our environment, in our situation here. In my house here, I don't have water from government. I don't have light from government. I have my own borehole. Do I have to sink a borehole in America? I have my solar. Right now, I am using solar. No, we don't have light for months. Do I have to look for solar? So these are situational. And if we don't wake up to that challenge, because life is all about challenges, solving problems, and evolving. We evolve from our environment and come to where maybe someday, sometime we won't need to do basic tests anymore. I think so. But for now, for anyone to supply drugs, even to government hospitals, we will need to check, because I know the kind of system we are operating in. And if we say no to all those caution, then that means we've not even started. It appears to me that we're not. So the question here again is, are we achieving recorded high results in our patient management from the pharmacist's contribution? Let us note again, for emphasis, that our contribution should start by ensuring that the drug that is being discussed or being used are original. They are what they should be. So requirement for this basic test skill, knowledge of physicochemical properties of drugs. Are these drugs acidic? Are they basic? Are they neutral? These are what we need to design 1 or 2 or 3 protocol step, whatever, to develop a basic test. If you have developed for one drug and I have developed, we can share notes. And if I've developed something I believe will help me identify one particular drug, or I can share it so that it can be adopted, it can be used far and wide. And if we join hands, according to our slogan, as men of honor, we join hands, then we're going to fight this battle and win it in no distant time. So what do we need? Solute-solvent interaction. Then different functional groups. The 5-nitroimidazole compounds, metronidazole, tinidazole, secnidazole. What kind of functional group do they have? Now, these NSAIDs that are acidic drugs, what kind of functional groups do they have? So what can I add to them that can precipitate them, that can do this, that can do that? They can be basic things. And we are pharmacists, we are chemists, we are pharmaceutical chemists by default. So every specialization that we have today, we need to carry from that understanding. So in basic test, we need this kind of apparatuses: test tube, funnel, beaker, volumetric flask. Simple things like this. Maybe 2 or 3 in every community pharmacy where we have a clinical pharmacist who wants results, who doesn't want to be an agent of peddling fake drugs in the name of I don't know. I didn't know. I got it from the right source. Source what? If I ask us a question this evening, that right source that you believe has given you correct drug, can you give your 1 million naira to that rep to vote for you till next month? If you won't run away. So why do you trust that something couldn't have happened, or I don't know? And so we need to take what we're talking about here very seriously. Let's move to the next slide. This basic test program, we put some relevance to us and to our practice, because we're going to be sure that we are using the right drug, we are getting it right from the beginning. And so therapeutic success is sure to come. This issue of safety is also sure to come, because these fakers can put anything. So if you have cases of kidney failure due to this or that, they say drug-induced kidney failure. Yes, the drugs we know can induce kidney failure. We know them. But even this one that we don't even know what they've put together, what they've packaged, can't it cause some atrocities and all of that? God forbid that it was actually a pharmacist that actually handed it over and wrote the label 2-3 times a day or that kind of thing. I think this thing should be done in the last century. We are living in a different world where a lot of knowledge is all over the place. And we must put all these things into perspective and design something that will make pharmacists of global reputation. So if we start doing this kind of stuff, even the biggest of consultants, pediatrician or oncologist, whatever, they will wait till the pharmacist comes or the clinical pharmacist comes to tell them that this drug is original. Because as we speak today, all of them, everybody is into drugs. Everybody is buying drugs. The consultant has his own drug. He knows who supplies him. And this one knows who supplies, up and down. That is how it is. The lab technologist or medical lab person also has his augmentin in his drawer, and they believe everything is the same. But until they know that a group of pharmacists who are serious-minded love to look at the authenticity of drugs before they are deployed into use, then they will know that they need to give us a little respect. They need to give us some salute. As it is that they think we're I don't know really, okay? So I can imagine when every healthcare team member start asking pharmacists, clinical pharmacists, are we sure the therapeutic failure is not traceable to? And then we can say something for sure. Don't just do, I don't know, we got it from the right source. Source what, man? My question here is that ACI, that lisinopril in stock, is it actually lisinopril? Or is just the label that we're looking at? So extension of clinical pharmacist relevance. When we start with basic tests and the little knowledge that we acquire from there, we can now start looking at this point of care test. Point of care test should be manned even by clinical pharmacists. Because just by the bedside, somebody has just been rushed to the hospital, the person has taken paracetamol with aspirin or whatever, and is mentally unstable, and there is vomiting or whatever. We can't just start hypothesizing. Maybe from the breath or from blood samples and all of that. Pharmacists can do all of these things. So clinical pharmacists in emergency care, in paracetamol poisoning, are we just going to be vomiting, literature until we start to put our hand to do one thing or the other to come up with this? That is when our relevance, not on a citing research output here and there. Let them wait for the pharmacist to come and collect one sample or the other or whatever. And then they're waiting for him to come up with results. It seems we have narrowed our relevance so much that if we're not there, things can actually keep going. We talk about therapeutic drug monitoring to our students every now and then. Even clinical pharmacists talk about therapeutic drug monitoring. Who is going to do this monitoring? How do we start? Is it the pathologist, the lab scientists that we still come and collect samples and go and test? Is that part of their? But we are looking for drug in this person's body. Whether the drug is up to the CP that is effective or not, I think it should still be pharmacists, if only we are ready to take up the challenge. So I think we should be ready. It can be I mean, if we think about all of these things, then we can come up with ideas that can make us more valued. So if you are talking about dosage optimization, it's not all about hypothesizing. We need to do one thing or the other, and all of that, and all of that, and all of that. So therapeutic drug monitoring is not rocket science. Basic text is not rocket science. We just need to expand our scope, our horizon, in the area where we become indispensable. So my suggestion at this point is that we open up frontiers in these areas for expanded practice and relevance. So without wasting our time, I know my time is far. Let me just try and round up. I have some practical demonstrations here. I should be rounding up in 5 minutes. For captopril, that's the first oral ACI. The basic test to know that this is captopril. Just take 10 milligrams of the powder tablet, dissolve it in 2 ml of water, add 0.5 ml of lead acetate, 8% weight in volume. A white precipitate is produced if it actually contains captopril. This can be done on a side bench, in a very small side, whatever, in any community pharmacy, or even in a teaching hospital, or any health facility by clinical pharmacists, or any pharmacist that is interested in quality. Because if the materials for our building is not of the right quality, then it can collapse. Then the second, magnesium sulfate. Example, magnesium sulfate injection in preeclampsia. We just remove the magnesium sulfate and start administering. We're not even sure whether it's actually magnesium sulfate or sterile water. It could be sterile water. And then when we administer and it's not responding, we are looking elsewhere. We are thinking spiritual or something. So how do we test that? Just weigh an amount of magnesium sulfate equivalent to 0.05 grams. Add water up to 5 ml, dissolve in 5 ml. Add 1 ml of hydrochloric acid. The 1 ml of barium chloride, 5%. A white precipitate is also produced. These are things that a secondary school chemistry person can do by instruction. But we can explain why these reagents, simple reagents in these small quantities, can help us know that this is what we have in our hands or not. Then phenytoin sodium. We say that's saturable kinetics, okay, and all of that, all those kinetic whatever. For us to know that we have phenytoin sodium, just open the capsule, and then we weigh out about 10 milligrams of phenytoin sodium. We add 2 drops of ammonia, then 2 drops of copper sulfate. We mix with a glass rod. A pink precipitate is produced. If the percentage you have is not pink, is blue or yellow, or no percentage at all, then we can start interpreting that either is a blank, just starch you have in there, or they put some other chemicals that we don't know the safety characteristics or profile of it. So I guess from today, before you hand that phenytoin sodium to that patient, or when that consultant, neurologist or whatever is talking about, we should know that are we sure we have the right drug? Then piperazine, 0.2 gram of piperazine citrate, add 3 ml of calcium chloride, boil in the test tube, it forms a precipitate. If you don't boil, the precipitate does not come out. Once you boil it, if you had acetic acid, just like 2 ml, the precipitate dissolves. This can be very interesting, I tell you. What are my suggestions? Integrating and highlighting the core sciences to clinical pharmacy. Clinical pharmacy is evolving. Every field is evolving. Every field is evolving. It must have a direction. So in which direction is clinical pharmacy evolving? We can't be doing the same thing we've been doing over and over again. We keep having the same results. We must have to do something a little different or add a little thing, and then we go, we get there. So a little clinical biopharmaceutics in my own field as an HOD in University of Uyo, I did some things. Some accreditation team came to my school and they looked for our manual. I gave them, are you sure you are doing things in this manner? Yes, we are doing it. I don't want my students to, they won't be able to hold a micropipette to pipette 2 ml of a particular level into test tubes. Nah. We are living at a post-genomic area. Biology has gone to the post-genomic area. We are not going to be doing pharmacy the way we've been doing over the years. Things have to just move based on the challenges we have around us. So expanding the scope of clinical pharmacist relevance, we will need to, I plead, I beg, let us put a consideration of what I'm sweating and pushing to us this evening. Finally, ladies and gentlemen, we have a research group in University of Uyo called Bioscientific Research Group. What do? We empower clinical pharmacy-based researches. When I say empower, yeah, I mean empower. You can imagine the different aspect of empowerment. We build clinical skills around community-based practices. We are talking about universal health coverage. And pharmacy outlets are more well-positioned to help achieve the vision of UHC in the next 8 years. But the truth is, we need to bring these community pharmacies together and reorient them and put them in so that things we can make an impact. So we build skills in support of community pharmacy practice so that we can actually occupy that primary healthcare position, and the challenges we can overcome them. So these are my references, ladies and gentlemen. Thank you for listening to me. God bless.
Yeah. Dr. Izzy, can you unmute yourself, please? Dr. Izzy, can you unmute yourself?
Thank you very, 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 very much, Dr. Sunday, my head of department. I totally enjoyed listening to you. I found this presentation very educative, very thought-provoking, inspiring, and motivating. And I see it as a clarion call to all clinical pharmacists, irrespective of practice setting. And I must really commend the passion in which you deliver this lecture. It did demonstrate your love for the profession. I'm happy I attended this presentation, and I'm sure many of us share the same view as I. So he has said quite a number of things, and I'm sure we have questions to ask. So before I ask mine, I will throw the floor open for others to ask their questions.
Let the speaker stop sharing his slides. Yeah.
Or if
Okay.
you can give your comment if you have one. Hello. Hello, we have about 58 participants here. I'm sure that somebody has something to say or a question to ask. I've seen some hands up here. Dr. Chinwe Udeka, Pharmacist Joseph Ole, Pharmacist Maureen Nwafo. I've seen it former Momo. Please, let's start with Dr. Chinwe Udeka. Please, can you ask your question or make your comments? Dr. Chinwe Udeka. Hello.
Doctor, can you unmute, please? Yeah, okay.
Hello, good evening, everyone. Thank you, Mr. Moderator. I hope you can hear me. Hello, can we all hear me?
Yeah, we can hear you.
Oh,
Yes,
okay.
we can hear you.
Okay, I start by thanking Dr. Sunday. It was a very, very detailed presentation, and the passion was there. However, as you were presenting, and I was so eager to get to the point where you would give us the practical steps we could use when we get to the lab. I was wondering, the drugs you gave us the basic tests for, I don't want to say they're not so common, but I was actually expecting that maybe we have an influx of anti-malarial medications in the markets. We have antibiotics all over the place. So I was actually hoping that maybe the basic test would have started with those very common ones, and we have them everywhere, and companies seem to just be going into their production. I mean, that would have helped rather than the not so common ones you mentioned. But all in all, it was a good presentation. If you have a reaction to that, I await. But then I also have a question. Just like you cited captopril, you gave us the step, the basic test for captopril, and I'm wondering, being captopril and ACI, can we also use that same step for lisinopril or any other one in that class, or is it just captopril? The basic test, is it unique and confined to each specific drug, regardless of whether the drugs are in the same family or not? That's one. Then two, when you brought up magnesium sulfate, I know in our hospital we have the injectable. So I was wondering, when you said to weigh 0.05 gram quantity, and it's an injectable, it's in liquid form. I'm wondering how that weighing is going to happen. How would I be able to weigh that injectable to get that 0.05 gram you talked about, and then to do that the other tests to get the results? Okay, I think that's about it for now. If any other thing comes to mind, I hope I'll be allowed to express myself. Thank you very much, sir. It was detailed. Thank you, sir.
Okay, I think we should take all at once then in our response.
Yes.
And so let's have a Pharmacist Joseph Ole for your question, please, or comment.
All right. Thank you. Good evening, everyone. I hope we can all hear me.
Yes.
Yes, sir, we can hear you, sir.
Yeah. So I am Dr. Joseph Ole, the chairman, SIPAM Pool of Expert. I want to thank Dr. Sunday for that great presentation. It's something that has given us food for thought, every pharmacist. And it's very, 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 very important and relevant to our practice. However, I want to say a couple of things. I'm aware that NAFDAC, under Dr. Dora Akunyili of blessed memory, developed something like this that was used to check fake drugs influx into the country, even right at the port then. And I remember that during my PharmD days at Uniben, we were taking through some of these things at the introduction level to PharmD. So there were those basic tests. We had that course under PharmChem. So they did it, and there were simple tests that you can do on the spot to just confirm whether what was being imported is what is actually in the thing. I also know that, but what is happening today, there was a gap when things changed, let me say, when Dr. Dora left NAFDAC and this thing fell into the hands of a pharmacologist. And we didn't do enough, like the physicians will do, to ensure that that remains in the hand of pharmacists. And I think from then, those important things waned off. Okay, so I'm also aware that in the National Drug Policy, it is a required service that when drugs are received into the hospital, I'm speaking from the hospital perspective now, that when drugs are received into the hospital, before they are released into the pharmacy for dispensing, these tests must be carried out, not just to even identify, but to actually test the quality. And because of the way the drug revolving fund scheme had been structured, and a system under which we have CMDs as emperors in the hospital, you find that they are not interested in that aspect. All they are interested in is how to fleece the EDRF money. And I say that with all boldness, and I can be quoted that I said that is what CMDs do. Okay, so that aspect has been neglected. For the hospital setting, I would say that we would remember that in 2020 during the lockdown, I made a presentation titled Unbundling Hospital Pharmacy Practice, and we in UITH, and we sent out the link. Some people joined from our side, and we extracted there to us what our hospital practice should look like. We delineated hospital practice into about 7 departments that should be under hospital practice. One of such departments is clinical pharmacy department, and then we have the pharmaceutical quality management and pharmaceutical products quality management departments. So in that structure, that department is supposed to have pharmaceutical chemists and people in pharmaceutics who will carry out quality tests regularly on the products that come into the hospital. Not even just that, but also the compounding, extemporaneous compounding that we carry out, because there is the science of solutions too that we may not, some of us may not be aware of, but we just use anything to compound. Sometimes you see some drugs are basic drugs, tablets, and in places I've seen them use vitamin C to compound them. I have seen where they have used vitamin C syrup to compound amino filling tablets, for example. And I was wondering, even if it's not happening, but it's a possibility that both of them are, you have a weak base and a weak acid there could be some level of reaction. So that department, we suggested in that presentation that that department should be recognized, created, and empowered with the necessary skill to carry that even before the drugs are sent to the dispensing area. I want to close by saying that while this service that's been presented is germane and very important to every pharmacy practice setting, whether community or hospital, I want to say that it should be handled with by professionals now, because clinical pharmacy is the core of clinical pharmacy is patient care. So, and really, in my place, with what we have done, we would not be so comfortable carrying out this test at bedsides. It should have been done and assured in before even the drugs gets to the dispensary. It is what every hospital pharmacy should be doing, and even the National Drug Policy makes provision for that. Thank you very much. Those are my comments.
Okay, thank you so much. Let's have Dr. Maureen Nwafo. Dr. Nwafo.
Good evening, everybody. Hope you can hear me.
Yes.
Good evening, ma'am.
Good evening, ma'am. We can hear you, ma'am.
Yeah, thank you.
Can hear you.
Okay. On behalf of our able chairman, Dr. Joseph Madu, I want to appreciate our presenter of tonight's clinical meeting. Thank you very much. We did a comprehensive work. In short, you have passion for that aspect of pharmacy practice. Kudos to you. Also, I want to appreciate our senior colleague in our midst, Dr. Lolojo. Thank you, sir. You're always there for us. Thank you. We are glad to have you tonight. I have few comments and questions. Tonight's topic is apps for us clinical pharmacy. And we all know that for us in clinical pharmacy, we have those that work in the hospital and those that work in the community. We are all clinical pharmacists, but our area of practice somehow differs. I want to start by saying that we are in the era of specialization. Whether you're in the community or you're in the hospital, especially those of us in the hospital. So we advocate specialization. For us who are clinical pharmacists in the hospital, as the last speaker said, you may not have time to start doing all these basic tests in our dispensaries. Before you get to your ward run, start testing drugs that you're going to dispense. So actually, this in the hospital is taken care of by the quality control units through compounding unit of the hospital. Their duty is to assess the quality of the drugs, not just the whether they are fake or not, but do they actually contain the actual quantity written on the package. So that's their specialty. And even in our college, we have faculty of drug production and quality assurance. So they specialize in testing, doing all these tests you mentioned, both those in the community and those in the hospital. So for us who are really in the core clinical area, like myself, I work in ICU. There's no way I can do all these tests you mentioned this night for me to, see my patient, do what and do all those things. Actually, for us in clinical pharmacy, you cannot do any pharmacotherapy, review without giving your patients quality genuine drugs. That's why we now have to go into specialization so that those in the quality assurance and drug production should do the dose tests before sending those drugs to us. So that's how we operate in the hospital. We may have a little bit challenge at the community practice. They can start this fine, those who can, but at least they can start from making sure that they get their products from the quality, from the rep of companies they know they are short of their quality, But it's a good one that will start from this if you can. Also, I want to say that we in the hospital, like inside the ward, we can just use the physical appearance of the drugs to say, okay, this may be fake or not. Okay, if you find such or you are not sure, you can still send them back to the quality assurance department to really recheck. I give you a scenario. Just last week in ICU, we ran short of noradrenaline. And I requested for more because we have patients who are in need of the drug. Then I was informed that our quality control department sent back all the batch we brought in the hospital back to the supplier, back to the company because they failed the test. I wasn't surprised. That's been what we used to do. So I went straight to the prescriber. I said, please, we don't have no adrenaline. See what happened. They failed test. So please, can we just use dopamine or adrenaline? And they were so happy that we can cross-check our drugs before bringing it into the wards. And they eventually used dopamine. So that's it. And also, you mentioned that when we were making our clinical presentation that we should start to talk about the isomerism, ionization, they're all fine, but it depends on what you're talking about. If you're not talking something like pharm chemistry or drug physicochemical properties, you're talking about case presentation of a diabetic patient, start to talk about isomerism and also, I don't think they all go well. That's my own view though. And I'm very happy when you mentioned therapeutic drug monitoring. It's a core duty of a clinical pharmacist. But we have, slight challenges here in these developing countries, It's a mandatory duty for us clinical pharmacists to do TDM before we give our patients vancomycin, aminoglycoside, those drugs that have a low therapeutic index. But the challenge is that we don't have the machine like HPLC, you can't do those things without those machines. It's not a basic test. It's not a simple test. That machine is very, very expensive. We are still asking God to help us to get one to our own center so that we can start doing the TDM. It's very, very important for clinical pharmacists to do before they give the next dose of those narrow therapeutic index medicines. And with pharmacists, we should open your British pharmacopoeia. You see those simple tests, even how to use just physical appearance to know that this drug may be fake or not. When you are in doubt, then send them back to the quality assurance department if you cannot lay your hands on those reagents that you can use to do those tests. Fine. So I want to conclude by saying that we are in the era of specialization, as I said earlier. Let's encourage pharmacists to go into specialization in quality control and compounding so that they can be really guided well to know how to do those tests. Then we in the world can actually get those drugs that I know that are genuine, that quality, that we can give it to our patients and be rest assured that are good ones. Thank you very much. That's my simple comments.
Moderator.
I'm done. Dr. Shets. Presenter, you can go on and answer the questions or the comments while we wait on the.
I think
Thank you.
is muted. Okay.
Can I go ahead and answer?
Okay, I thought to myself.
Okay.
Eshet is back.
Thank you, Dr. Omar, for if you have more time. Hello, sir. Yes. Please do.
Can you go ahead and answer
Hello,
some of
sir.
the questions already raised before more can?
Okay, I can go ahead.
Yes, advice should do. Thank you.
Please, sir, go ahead. We are waiting for you to go ahead and answer the question.
It's okay. Thank you very much. I appreciate the compliments. I'd like to start with the first question. The question was asking about this basic test for the common drugs, antibiotics, and all of that. I must say at this point that everybody believes that the way you speak and people can smell ampiclox or the penicillins from your breath or from the urine is no longer what we have. So the truth is, what we have, whether you call it bicham or whatever, is only God that can help us. We have some basic tests already developed for some drugs, some common drugs, like Flagyl and the 5-nitroimidazole compounds. The 5-nitroimidazole compounds have the same functional group. And so the test we developed will pick any of the metronidazole, secnidazole, tinidazole, and all of that. What it is actually looking at is the azole entity that all of them carry. And then the 5 position has the nitro. So those are the things that make it very sensitive. Okay. Now, some drugs like captopril, enalapril, lisinopril must carry the same, if they are developed from the same backbone. Only some methyl group remove here, all those structural, whatever, modification, all of that. Yes, they can still have but some drugs may not have the same backbone, and they may not. So what we need to do in this aspect is to sit down and look at what they present and come up with a position that our colleagues can use. We are not saying all of us must start developing basic tests, but whatever we have developed, we should be able to adopt it. We should be able to give a framework in our system so that those who are in the ward, the ICU ward, for example, can lean on their, we can't be jack of all trade. All we are saying is we need to know that fake drugs are just all over us. They are overwhelming us. And for us as clinical pharmacists to have results, then we must call our colleague who is there. Look at what we are seeing. See the outcome we are having here. I will look at it. I'm happy that noradrenaline was returned. That's great. I'm so excited to hear that. And then something else was used. In other clients, we'll continue to use that noradrenaline. And a lot of stuff will, and as a pharmacist, I'm not happy that things are that way. So I think I've answered the question from the first inquirer, that one test may not carry for the entire group unless they have the same backbone, they have the same skeleton or the same, like the fluoroquinolones. Okay, they have a fluorine group at a particular end, and they may show the same attributes. And because we are pharmacists, yes, I know we have specialized, but we need to be sure that we have a knife that is very sharp to cut this tree that we are cutting. If the knife is not sharp, then we're going to have problems. So whoever is the goalkeeper should be able to stand and defend us. When I was growing up, I want to play football. All of us want to be the striker. Nobody wants to be the goalkeeper because it appears things are happening out there. And so a small shot that shouldn't give us a goal is what somebody will shoot over our heads. And all of us are running after the ball, and the ball will stroll into the... So we must put our house in order. I guess that is because of time. Let me move to the second one. Yes, I appreciate those comments. And I want to thank God for Dora Kinyuli. Let me just say, working my way towards getting to that position in NAFDAC. And that's why I just qualified as a public analyst to analyze food, drug, and all of that. Because if we say we want pharmacists to be there, most pharmacists are not going to, qualify. When we're doing the qualifying training in Institute of Public Analysis of Nigeria, there were only 2 pharmacists there, one from OAU and myself from University of Uyo. The people who fill the whole place are chemists, biochemists, microbiologists, and now I have a seal from federal government backed by Decree 100 or something, that if I analyze the drug, I can stamp on that drug and sign it. And any judge in any court, see that seal, use that to judge based on what I've recommended. So we pharmacists, yes, have to put all of these things in place so that we can sit in that NAFDAC. But they're not going to give us that seat because we are pharmacists. We must show that when it comes to analysis and all these things and the passion to make well, I hope that works because Nigeria, they actually know what they want. But we who are sincere, who want results, we are the ones that are dispassionate, and God will help us. So yes, all of us cannot be in the lab, but do you know sometimes when people have emergency like that ICU, somebody has just come in there, the person is vomiting, has just taken drugs, overdose or whatever. As pharmacists, we can actually take samples from there. Well, we may ask our colleagues to come from the lab and take the samples and go and analyze for us to have an idea what is the drug that is causing this whatever. I don't want us to allow chemists and whoever to do that. We pharmacists, we are talking about drug poisoning here. And even if it's not drug poisoning, even if it's some other chemicals, all our knowledge of toxicology and everything, the truth is we need to guard our house fully well so that it's our colleague, is a pharmacist that is still doing this. But if I just say, well, I'm a clinical pharmacist, I'm going to only be at this point, somebody else may do that. Somebody else will do it. More so if we don't have a structure in place in our system where those things can be done. So the point of care actually happens in the ward, in that ICU where you are, where the person is lying down on the bed. That is where the analysis, or I'm sorry, the sampling is taken. We may not do it there, but that's still a part of what we can put to our domain. Okay, so yes, all of us will not do everything. We can have people in core clinical areas, but we are saying that even those are the core clinical areas, we must tell, we are like strikers, people like Ronaldo that scored the goal. Then there's a goalkeeper, so that we can actually win the trophy on the long run. If Ronaldo is just there waiting to score goals and the back is very weak, I'm sure those football enthusiasts can analyze that better for us. Okay, if you say reps from company, people are really compromised today. People are really compromised. People are really compromised that even reps can, who are even the reps we are talking about today? Some of them are not even pharmacists. I've interacted with a lot of them. They're microbiologists, they're computer scientists. Oh God, I don't know what are we really talking about? And these microbiologists or whoever who are now peddling even prescription drugs, what are they looking for? Money. So if somebody intercepts them and start talking business as usual, are we sure that, well, I don't know. But we need to guide our back and be sure that our work gives a results. That's what we're talking about here. Then, to the comment on isomerism, physiochemical properties, I want to stop a bit and make a point here. If all that we say in a grand round with the medics, the lab scientists, and all of that are not things that are more core on drugs. If we don't talk from the angle that is our exclusive domain, and we're not talk about pharmacology, how phospholipase C, how it breaks we're not going to all those. They know you're not saying something new. What can only be new to them? Tell that medical doctor that metronidazole is a basic drug. It has a PK of this or that. If you leave all those ones out and you only talk, or the excipients that they have used in this drug and that one they have used in that drug, you think is for pharmaceutics alone. In their drug selection, maybe you are getting it wrong because you didn't select the right drug that has the right. We need to expand our horizon and not be so narrow. When we are so narrow, people can capture you. They know all that you are saying is what they also know. So what are you? What is new? That is the point you are making here. I will only listen to you if you have something new to tell me. And what is new? You are coming from the pharmacy end. Then you must be able to tell us what is native to, otherwise, all that you are saying is what that consultant ENT or consultant neurologist, so it doesn't even think you are contributing that much when you are not around. I don't know. So I think I don't want to overflow that. Yes, the pharmacopoeia. If we go to the pharmacopoeia, there are a lot of simple text already there about most of these drugs. So we can stem from there and we can develop our own. I developed on 5-nitroimidazole. I presented that one in Berlin, Germany, 2007. The Viagra was my first time of traveling. That was 2006. I presented on Viagra, and Pfizer was there to listen to what I had. And what I needed to do is to start putting my chemicals on strips so we can add it to the drugs, about 4 or 5 strips. So when you remove one tablet, you can sacrifice it, and we tell you 1 or 2 things, even for the public to. So these things are going to, even if the public will not see pharmacists, watching their back, somehow, everybody is interested in quality. Nobody wants to take a spurious drug. So the captopril I mentioned earlier has a chiral center. And so the chemical that they have used to prepare the captopril supply to you, is it the D or is it the L? Some time ago, Pfizer came up with amlodipine, and it was 10 milligrams. Before we now came to know that amlodipine has a chiral center. So we now have S-amlodipine and R-amlodipine. And now we don't even need the R, we need the S. So science is evolving. So as pharmacists, you are the one that will tell them that S-amlodipine is what we need because of the enzyme specificity and all of that. But if you say we shouldn't go to isomerism, we shouldn't talk about that. When we say case study, it should be pharmaceutical case study. We should be talking more about drugs. I've attended one seminar like that, and everything they were saying were more of what a medic will just, they won't be glued to their seat listening to you. They can only listen to you if you can dissect and open up drugs the way when drugs are taken with a fatty food or oily food or empty stomach or this or that, this is how so they can tell their patient. So sincerely, I think our case studies should have the flavor of a pharmaceutical more. Otherwise, we're not making a lot of So I think with that, I've answered most of the questions because of our time. Thank you very much.
Thank you, sir.
Hello. Yes. Hello.
Hello, can we have Nuruddin? Thank you, sir. Can we have Nuruddin before we run off? Dr. Nuruddin.
Thank you very much for the opportunity. Am I on?
Yes.
Yes, you are, sir.
Mine is not also a question, but some observations. I think a lot of people have spoken, and I want to start by commending the presenter for this wonderful job. And I want to see that when I first saw this topic, basic tests in clinical settings, actually, I must confess that right from the very beginning of this presentation, I was just asking myself whether we are really doing a presentation on basic tests in clinical settings. I was thinking, when you talk about basic tests, if really this is what we're going to talk about, it would have been basic quality control tests, because in clinical settings, basic tests is beyond quality control, just like some of our previous speakers have said. Quality control test is usually done at different levels, right from NAFDAC. Let me even say that Dr. Joseph Ole mentioned something about NAFDAC during Dora Okwili, and at the end of the day, he ended up saying that NAFDAC has derailed. But I want to inform the House that as at last week, NAFDAC has even grown 100 or 200 times higher than when it was during Dora Okwili, because I think that last month, NAFDAC was issued maturity level 3. That's to show how much standardization has gone through in terms of authenticating NAFDAC. And most of the quality control tests mentioned in this particular presentation are usually quality control tests that are done first at the pharmaceutical company, secondly at the regulatory body, and thirdly at the institution at the point of delivery, probably during the delivery in the hospital. And of course, just like other people have mentioned, these are kind of tests that are done before even a drug is accepted into any institution, whether community or hospital. It is unfortunate that a lot of hospitals have neglected this part because of the reasons so far mentioned, but we must remember that a lot of hospitals are still doing it. One of the reasons why I say a lot of hospitals are not doing it is you can remember the case of my Pekin. The reason why the incident of my Pekin happened was because there was no validation or authentication before drugs are collected. But for me to hear that this type of tests are being done at bedside or on each patient, I think it is actually a delusional thinking. When I begin to think that I will be testing for, I will be doing quality control tests on every patient in the ward. When I go to the ward and 2 drugs are given to a patient, let's say the captopril, you give example, and spironolactone. These are common drugs that are found in a single prescription. One of the ways I can, not necessarily myself carrying out the test, but EUCR alone, when I look at the results of EUCR, can give me an idea whether there's hyperkalemia or otherwise, because each of these 2 drugs can cause hyperkalemia. By the time you combine these 2, this is not doctor's work. This is pharmaceutical work. A doctor looking at a patient under this type of combination and the manifestation of hyperkalemia will think there's another disease, that he is bound to prescribe another drug. So the role of a clinical pharmacist here is to identify that based on the laboratory test done by a laboratory scientist, not necessarily a pharmacist. there is hyperkalemia, and this hyperkalemia could have been as a result of this combination. So how do you go about it? It is either you reduce the dose, you stop one of the drugs, depending on general discussion. And that is the aim of clinical pharmacy study. So when we continue to buttress the point that we must not focus on, that, what we are trying to do in clinical pharmacy is to elaborate and strengthen pharmacy so that we create roles. Tell me, a consignment of drugs is being received by a hospital in a year 3 times, 4 times, or 5 times. How many pharmacies do you need to sample drugs and do this type of basic test in a hospital just to confirm? Do you need 5, 10, 15? How many pharmacies do you have in? This is one of the reasons anytime there's going to be employment or recruitment in a hospital, the doctors feel you don't have a role. When there is need to recruit 100 staff, they think pharmacy should be given 2 or 3 because this is what we want to be doing. But by going into clinical settings, creating value, looking at the drugs they are combining on patients, you are creating value for yourself and you are creating relevance such that anytime, any moment, any clinician would think about pharmacy and would think about pharmacy because you are adding value to the patient. But going to the patient and carrying out basic quality control tests, which you have, the presenter has said it clearly, that you cannot sit down here and tell me that a professor of pharmaceutical chemistry or someone who has a pharmaceutical chemistry who does not have pharmacy as a base cannot do this test. Or somebody who has done microbiology or another course and has gone to pharmaceutical chemistry to do master cannot carry out this type of test. We are only deceiving ourselves. So if you think in the other clinical setting, we are competing with doctors. If you go back to this area, you are also competing with other people who are not even professional. So it's a matter of we must embrace all and we must widen our horizon to hold on to every part. And what clinical pharmacy has come to do now is to create roles for pharmacists so that we do not put ourselves at the back of the hospital. We must be core members of clinical teams. So we must continue to push for that, and we must continue to encourage our colleagues to be part of clinical pharmacy team and to be part of the delivery team. Thank you.
Okay, thank you, Dr. Nuruddin. I think that should be the last comment for today. So can the presenter wrap up, please?
Thank you very much. I appreciate the last speaker. We won't have to go to the bedside of the patient to carry out our basic tests. Like I said in the discourse, that this basic test becomes important because it is situational, it's environmental, and we see it affecting our results. I'm aware that in some institutions, their prescriptions fly out, and the drugs that these people are actually using might not even come from that hospital. Some of them even think for their own reasons, whether cost or whatever, may not even buy their drugs from so drugs just fly in here. So all we are looking at here is, are we getting results? Are our results due to spurious drugs? Fine. If we have a controlled, like in America or developed countries, their drugs, you are sure of where the drug is coming from. And so if it's not right, then you know it has to do with maybe the patient. But now it might not be the patient, it might be the drug in Africa, where in most hospitals, you are not even sure where the drug that person, that spironolactone and the captopril this person is taking. We don't even know yes, I know we need to, those are 2 drugs that can cause but yes, I'm okay with all of that. But we're not looking at how many of these stuffs are due to drugs coming from different angles and all of that. So I wish that all our universities, teaching hospitals, general hospitals, community practice pharmacists can actually put their hands on deck and ensure to the best of their abilities that the drugs they have, the drugs they are giving out are of the right quality. So at the bedside, if we are vast with knowledge about this little, we can as well still broaden our horizon on point of care, because there are cases where in that ward, you are the in charge in that ICU. And look at the case we are talking about here is a case of overdose of a particular drug. What do you do this time? We're not talking about stories now. What impact do you make there? So we must still come back to all our, and then maybe it might not even be you. You can call your colleague who is a teamwork but as a team, let us bring drug quality as a major consideration, because that is what determines many things about our practice. Thank you very much.
Thank you. Thank you, sir. You have done very, very, very well. Our time is very fast spent. We are just 10 minutes to 11 p.m. So I think we should wrap it up. Thank you very much, sir. So I will respectfully invite Dr. Ibrahim Bello to say the closing prayers.
Okay, can we pray? We give thanks to God Almighty for making this special edition of our clinical meeting a very great success. We thank Him for the knowledge that we have gained tonight. We only pray for the speaker who has imparted us greatly knowledge, that God Almighty to continue to bless you and increase you in His knowledge. We thank everybody that have come tonight. We pray to God Almighty to be with us, to bless us, and to bless people in general. Amen. End of prayer.
Amen. Amen. Thank you, sir.
Amen.
Thank you.
Amen.
We have come to the end of the meeting. I'm sure we have learned quite a lot today.
Amen.
On behalf of, thank you very much. Thank you, sir, for the wonderful presentation. We are very happy. Thank you, sir.
Good night, everyone.
Good night.
Everybody, good night.
Good night.
Sweet dreams. Good night.